This is the lab lecture that goes before you do the cell division lab, which is what you all have scheduled to do on Tuesday. So I think it is definitely going to help get ready for this lab. If you watch this video, if you also read through the section in your textbook about cell division and mitosis, and that will help you be much more efficient in the lab, and you'll know what you're looking for. It's just going to be extremely helpful. So now that we've looked at the cell, we've looked at the nucleus and the organelles, we've talked about things moving in and out of the cell, and we've done all of this without really talking about what one cell does kind of in its little cellular daily routine. So that's what's going to come up next, and that is called the cell cycle. So we're looking at just one cell, and if we're looking at kind of its, again, daily routine, not that this happens on an hour or 24-hour cycle, but what a day in the life of a cell looks like, that is called the cell cycle. The cell cycle has 4 main sections, and this picture makes them look like 4 equal pieces of the pie. I don't know that that's accurate, but okay. So the cell starts in what's called G1. G stands for gap. Although I will tell you, I usually think of G as representing growth, because in the first gap phase, the cell grows. And then it says normal metabolic roles. That means whatever normal cell function, whatever reactions are normally occurring in the cell, if that means the cell is breaking down glucose for ATP in cellular respiration, or if the cell is producing a certain kind of protein, which we're going to get to, whatever normal reactions that cell needs to be doing, they're doing that in G1. That's kind of just its normal going about its business. Then the next phase is called the synthesis phase, or S phase. And this is where something very special happens in the nucleus, and that is called DNA replication. DNA replication is a fancy way of saying that the DNA copies itself. And we're going to look at that process in more detail. But this happens somewhere in the middle of the cell cycle. And the most important thing that you need to know is that this has to happen before the cell divides, which we're about to get to. So the DNA has to copy itself before the cell divides. And this is so that there's enough DNA for 2 new cells, because once we have 2 new cells, we want them both to have a full set of DNA. Then the next part of the cell cycle is called G2. Again, I like to remember G for growth, because the cell is still continuing to grow in this phase. Now we're going to talk more about each of these in the normal lecture video. This is just kind of a pre-lab video to kind of get us ready to know what we're looking for. So we'll go into more detail about the cell cycle in the regular lecture video. All of those 3, G1, S, and G2, are together, lumped together, called interphase. And when we look at them under the microscope, they all look the same. We can't tell if a cell is in G1 or G2. It's just going to look the same. It looks like it's in interphase, and we'll show you what to look for there. Then the excitement occurs, and this is what you're going to focus on in your lab, is called the M phase, and M stands for mitosis. And then you see that section of the cycle is broken up into 5 different sections. And so we're going to spend most of our time here, because that's what we're going to look at specifically in the lab. But this is the process of the nucleus dividing and eventually the cell dividing as well. And then once the cell divides, it goes back into G1 and starts that whole process again. So any cell anywhere is at some point in one of these sections of the cell cycle at any given time. This just goes through basically what I said in a little bit more detail. Gives you a little more information about the G1 and G2 phase. We're kind of vague on these. Again, when we look under the microscope, we're not going to be able to see, oh, hey, we're in G1. But it is important that you know these phases in order of the cell cycle. Something to notice about G2 is it talks about repairs DNA replication errors. We're going to talk about that when we get to DNA replication. And it also kind of prepares for cell division. It talks about making enzymes that control cell division. So G2 is growth, but it's also kind of getting ready for that main event of cell division. And then, of course, here is the main event, the M phase, and this is what we're going to look at in way more detail. It is mitosis. Mitosis is specifically the division of the nucleus. Okay, this says cell replicates the nucleus. And really, that is what happens, because we're going to make two nuclei from one. Then right after two nuclei form, then the cell itself splits in half and making two new daughter cells. They're always called daughter cells. The cell cycle duration varies between cell types. So when I was saying that it's not necessarily a 24-hour cycle, not only that, but it also varies between cell types. So if we're talking about a muscle cell different than a skin cell, their cell cycle is going to vary. Approximately, a cell is spending 78% of its time in interphase. So that's G1, S, and G2 all together. So when you look at your sample under the microscope, you are going to see most of your cells are in interphase, about 80% of them. And that's because if we're looking at any given sample, we would expect a cell to be in that phase for 80% of the time, 78%. And so when you look at your sample, don't be surprised that you see lots of cells in interphase. Okay, then it goes from there. We're going to look at prophase, metaphase, anaphase, telophase. I know you may not know what those mean just yet, but those are the 4 phases of mitosis. And this gives you a breakdown. 14% of the time is in prophase, and then so on. And you see anaphase is real short. So you may, during the lab, have to look around a little bit to find a cell in anaphase. That's okay, but you still need to find one to be able to see what anaphase looks like. Mitosis is cell division, and it happens in all body cells except for the egg and the sperm cells. We will talk about meiosis is the process that makes egg and sperm cells. We will talk about that in a separate lecture, but mitosis is what you're going to look at in the lab, and that is what's happening in all body cells. So your skin cells, your muscle cells, bones, all of your body cells are going through this process to make new cells. And remember back from the cell theory, we said that all cells come from pre-existing cells. Well, this is how it happens. This mitosis process is exactly how it goes. Some reasons that a cell might need to make more cells, not only growth, but that's a big one, but replacement of cells that die, and then repair of damaged tissues. So if there's tissue damage, the cells want to regenerate and repair that. Then cell division and mitosis is how that occurs. Mitosis has 4 phases: prophase, metaphase, anaphase, telophase. You could remember PMAT if you want. That might help you remember the order that they are in. Okay, this picture goes through each one. I'm hoping there's a slide for each one individually. And yes, there is. Hold on, I just want to make sure there's a picture. Okay, good. Okay, so we're going to go through this slide. Is going to be broken up into the 4 phases in more detail. So, but this is a good kind of overview summary of what happens in the process of mitosis. So the first step of mitosis, or the first phase, is called prophase. The chromosomes, and that's our DNA, during mitosis, the chromosomes form. And we've talked before and called it chromatin. Well, during interphase, it looks like chromatin. It just looks like kind of a big ball of spaghetti. In the first phase of mitosis, the chromatin condenses. It kind of tightens up, shortens, and thickens, and it makes it more visible to us as chromosomes. Okay, now we're going to see lots of pictures of chromosomes like an X shape. Really not going to look like that. But it will be able to distinguish one chromosome or a chromosome from each other. Instead of looking like a big ball of spaghetti, it's going to look like a big ball of worms. Now, I know you're thinking that is not a big difference, but when you see under the microscope, you'll be able to tell what I'm talking about. We're going to talk about what we're looking for at each cell stage. The reason that chromosomes need to form is because during mitosis, we're going to organize and separate these chromosomes so that the 2 new daughter cells get the right amount of chromosomes. And so it's easier to distribute DNA when it's in chromosomes versus chromatin. So in chromatin, it's just a big old ball, but in chromosomes, they're kind of organized a little bit better. So 46 chromosomes for humans. That's 2 chromatids. A chromatid, if you're thinking about an X shape of a chromosome, a chromatid is one side. Now it looks like this now because it has been replicated or copied during the S phase of the cell cycle. So those 2 sides of that chromosome are similar but not identical. They hold the information that is similar, they hold genes that are the same, but we'll get into that as we go on into genetics. Also during prophase, the nuclear membrane disintegrates and kind of goes away or disappears. Also what happens during prophase, you won't be able to see this, but the centrioles start forming the spindle fibers. And you won't be able to see this during prophase, but you will be able to see it later. But spindle fibers grow towards the chromosomes during mitosis, especially in prophase. The spindle fibers are important because they're going to basically guide the chromosomes where they need to go during mitosis. So there's a picture on the left of a drawing of what it's going to look like, and then the picture on the right is more like what you might actually see under the microscope. I tell students that if you see a nucleus that has some white dots in it, that is prophase. And those white dots are basically the space where there used to be chromatin, but now it is condensed into chromosomes. They are not going to look like cute little X's. I hate to break your hearts, but they are not. So if you see a dot, which is a nucleus, and within that dark space you see some kind of light dots or some light space, that is going to be prophase.